so much, and thank you for the opportunity to get started here. I'm excited to share a project that I have going on. This is a new bit of research that I've been working on, looking at some antiviral properties of eucalyptus leaves to hopefully help our koalas. So for those of you that know me a bit more, my focus has been on a virus that's been affecting our koala populations here in Australia. It's Koala Retrovirus. And it's quite ubiquitous in our koala population. So in northern Australia, and so in Queensland and New South Wales, all of our koalas have this virus. Whereas down in southern Australia, so in Victoria and South Australia, we're finding more and more koalas are testing positive for this virus. So it's becoming a major problem because we're finding when koalas have this virus that we're seeing increased rates of chlamydial disease, which is their major kind of infectious pathogen. We're finding more incidents of cancer when we have high levels of this virus. And we're starting to see more immunosuppressive conditions that some veterinarians have started calling AIDS-like syndromes when koalas are expressing this virus. So I'm part of the research group with Peter Timms, and a big part of our research focus is looking on vaccines. So we are currently working on a vaccine for CORV, or Koala Retrovirus. But at present, there's no treatment for koalas that already have this virus. And so that's something I'm very interested in changing. So when we think of treating a retrovirus, the most common example we have is HIV. And to treat HIV, we're very successful with drug cocktails. So there are various compounds that can affect the entry of the virus, the reverse transcription, because retroviruses are RNA viruses, so they have to be translated to DNA, so we can inhibit them at that step. These viruses then need to integrate into the host genome, so if we can inhibit that integrase activity, we can prevent the virus from spreading and continuing. And then finally, as the virus is exiting the cell and maturing its polypeptides into mature peptides, we can also inhibit the virus at that step. So there's several very known steps where we can get at this virus with drugs and compounds that we know work for retroviruses. So our question became, what could we do for koalas? Because it's not really practical to give koalas out in the wild drugs every day. And the obvious answer is, well, can we target something and can we find something that they already eat and somehow try to introduce that to koalas more often? So for those of you that know our koalas quite well, koalas eat eucalypts as their main food source, and they're quite a diverse group of plants, and shrubs and flowering trees make up the eucalyptus family, with over 900 species currently characterized. However, koalas only eat about 10% of these, so about 90 to 100 species of eucalypts are known koala food sources. And we do know that a number of eucalypts contain medicinal properties. So this makes it a really promising avenue to see, well, can we find some antiviral properties in these trees? So this particular project goal was to look at some koala food source eucalypts and see if we could identify some antiviral potential. So we've partnered with the Gympie Regional Council to look at some local trees, and this has been a fantastic collaborative project with our plant biotechnology team here at USC, led by Steve Ogborn, along with bringing in some expertise from down south at Western Sydney, and some current Koala Action Inc. is currently interested in helping us with this project as well. So to begin, we picked we needed some trees to look at. So we partnered with Gympie Regional Council to get samples of eucalyptus trees that are known koala food sources here in the Gympie regional area. So we ended up with we targeted 9 different species of eucalypts that are primary food sources, and 10 eucalypt species that are secondary food sources. So you see we get a number of gray gums, red gums, ironbarks, that we know the koalas in this area do eat. Added to our list, we wanted to add a particular species of eucalypt that we knew that has been published to have antiviral potential, and that's white stringy bark. And that's typically found kind of in the Sydney to Melbourne area. So it's a southern particular eucalypt. And so we know that it has potential, so we included it as a positive control. And so we went out and to find samples of these trees to see what we could figure out, see what we could find from them. So to look at this, we wanted to get the compounds out of these leaves. And to do that, we used a range of 4 different solvents. So We're trying to dissolve all the different compounds out of leaves. We know that from our basic chemistry that not every compound will dissolve in one particular liquid. And that's because different compounds have different polarities, different solubilities. So for this project, we used a range of 4 different chemicals, ranging from hexane, that has a very low polarity, up to water, that has a very high polarity, to try to dissolve out all the different compounds that are present in the leaves. And so we think we've covered quite a reasonable range. And although we have an idea of what kind of compounds are going to be in each extract, we won't know for sure until we do more specialized analysis. And so then we have to figure out what's causing our specific activity will require further analysis down the road. So to give you a quick idea of what we did, we took our leaves and we ended up having 44 leaf samples when we were done. And we extracted them with our 4 different compounds, or 4 different solvents. And so basically it's mixing it in and then taking the compounds that dissolve in that solvent, drying them out, and dissolving them in a final solution of DMSO before we test them. So that's one thing I wanted you to be aware, that when I do my antiviral testing, all the compounds are in DMSO or in a DMSO water solution. They're not in the hexane or the methanol or the solution we use to extract them. The other little key bit of chemistry to know is that chlorophyll, which gives leaves their green color, will come out in these 2 extracts. And so because of the chemistry, the particular tests we're going to use for our antiviral testing are colorimetric. We had to remove our chlorophyll before we could test them, so they got an extra step to remove the chlorophyll. But it was basically to create these crude extracts, so then we could go on and test for antiviral activity in these leaves. So happily, there are commercial kits available, particularly for HIV drug discovery, and we have evidence from our collaborators that compounds that work against HIV also work against Koala Retrovirus. So results that are positive in these testing have a very good chance of being active against our koala virus. And so what we've done, I started off with an anti-integrase test. So that was that 3rd step in the life cycle. And we tested our 4 extracts and all of our different samples in this assay to see what inhibited this viral enzyme the most. So what we're looking for is that we're hopefully going to have the most activity, so highest on these graphs, farthest to the left at the lowest concentrations. So you can see here that what I've circled here in red are the compounds that ended up having, that had the most activity, the most reproducible activity at the lowest concentrations. So zooming in on that particular extract, it found out that the methanol extract gave us the best anti-integrase activity. And this wasn't surprising for us, as in the literature, when we do a lot of drug discovery, we do find that often the methanol or ethanol extracts of plant compounds tend to have the most antiviral activity. And this is also where our positive control, our white stringy bark, our known antiviral compound, glomboidian A, is found in a methanol extract. So this gave us a lot of confidence that we're getting on the right track and we're seeing compounds that have activity that other groups have seen. And when we zoom in on what these actual dots are, we actually found that we had 7 species that showed greater than 50% inhibition of our enzyme at 100 micrograms per mil, which was the lowest concentration I tested in this particular study. So we found we had 4 of our primary koala food species, or our leaf species, that had this activity, and 3 of our secondary. So the gums came out well, along with some of our black butts and mahogany. So this was really promising. I also had access to an anti-reverse transcriptase assay. Unfortunately, in our current COVID situation, there were some shipping and receiving issues, so I couldn't get as many tests in as I wanted, but I was able to at least test 3 of my extracts with a few of my leaf samples, and I was able to find that, as you can see, we do get activity across the spectrum, and there are some samples that have some really positive results. So zooming in again, it was again the methanol extract that had the best activity for this reverse transcriptase step, so that 2nd life cycle of our virus. But unfortunately, this is still quite preliminary, as I need to confirm this with more testing. But interestingly enough, when we zoom in as to what trees and what leaves produce this best activity, it's actually a different list of species compared to the ones that had the best anti-integrase activity. So here again, we have two primary leaf samples, or primary food sources, sorry, two secondary food sources, and actually our reference, our white stringy bark, actually had very good anti-reverse transcriptase activity. So this wasn't surprising that it's a different list. Again, it's a different enzyme and compound to target, so we would expect different compounds to have effect. So this again gives us some confidence that what we're finding are very promising and very hopeful results. So in conclusion, we found that, as if you noticed on those graphs, there was lots of dots in and around that graph. So we actually found that all of our eucalypts had some level of detectable antiviral activity, and that our methanol extract showed the strongest activities for both of the assays that we looked at. So that indicates an extract that we should be following up on the future and narrowing down our future work. And that we did have different species that had the strongest activities in each assay. So this narrows down our big list of 19 down to a smaller list that gives us some future areas to look at. So just very briefly, things that I'm going to look at continuing in the future. Obviously, a lot more chemistry is needed because we don't know what compounds gave this activity. And this is, of course, a very small sampling set to see if this activity is more widespread in these particular tree species. Obviously, translating these results directly to our Koala Retrovirus. As I said, these kits are based on HIV enzymes, so determining how that works with our koala virus. And finally, we actually have a fantastic opportunity because eucalypts are actually natural food sources for koalas, and this is what koalas normally eat. We're actually in a fantastic position to actually do feeding trials and actually be able to offer koalas diets with these particular leaves and see if we can actually have a direct effect on the virus loads that they have. So this is a kind of fantastic advantage that most drug discovery, especially for humans, doesn't have the opportunity to do. So I'm very keen to move forward with that and see what we can do with that. So of course, a project like this has lots of people involved, so I want to thank everyone in our chlamydia and corv research group and the natural product chemistry group, our collaborators both in Western Sydney and all of our koala partners that have made this possible. And so I thank you for your time and I'm happy to take any questions. And I don't know if it's best if I stop sharing my screen, if that makes it a little easier. And so I don't know if it's easiest to have questions in the chat or if questions any other way.
So first of all, I want to thank you very much, Bonnie, for a great presentation. And I think the work is beautiful. The whole koala project has just been amazing. And I think for me, it's particularly lovely to see how, you know, you've taken it to interacting with Steve and the chemistry group, which, you know, is a nice example of how when you are in a center where you know about each other's expertise, you can, you know, take it to the next level and incorporate it in your research. Naturally, we're running a little bit late in time, but are there any questions now for Bonnie? If yes, if you can write them in the Q&A panel, or does anybody want to have a specific question now? If not, I'll just have a quick one, Bonnie. When you look at the amounts, and I know this is still, you know, early days for you, but when you look at the amount of the inhibiting compounds in the leaves compared with what a koala would eat, is it roughly in the scale?
That's something we're not sure of yet. That one thing that that's kind of our big next stumbling block is the fact that koalas have developed quite an extensive range of liver enzymes to deal with most of the toxic compounds that are in eucalyptus leaves. So although we can find antiviral activity in the leaves directly, our next big challenge is to see, does that activity get through the koala liver and become a circulating compound, which is where a feeding trial creates a fantastic opportunity to see if we can actually see an effect. So we're not sure, but stay tuned. That's next on our list.
Fantastic. Well, Bonnie, thank you very much. And I would like to, great start, and I would like to now invite Volker for his presentation.
Thanks, Abigail, and thanks, Bonnie, for a really nice talk. Now from cute and cuddly, we jump straight into creepy and crawly. I just start sharing my screen. Okay, can you all see my screen now?
Not yet.
Oh.
Did you press share?
I did press. Hang on.
Yes, now we can see.
So is it a write mode or do you see the presenter mode?
We use the we see the presenter mode. You might want to go into the...
Okay, I'll swap the displays. Is it better now?
Yes, thank you.
Okay. Yeah, so I won't be presenting a specific project. I rather give you sort of the master plan of how I plan to have my lab set up, because I just started this year, so I don't really have any specific project that I finished yet at USC. So we'll dive straight into it. So my research is obviously looking into arthropod venoms and how we can utilize them for novel therapeutics or bioinsecticides or molecular tools. And not all of you will be familiar with venoms and venomous animals. So I'll give you a little bit of background on that. So venom usage is actually not rare in the animal kingdom. So if you look at the animal tree of life on the left, all the red lineages, they actually contain one or many venomous representatives, whereas the black lineages, they don't. And so overall, you can see more lineages actually have venomous representatives than those that don't. And if in terms of percentage, it's about 15% of all animals actually use venom. So that's about 220,000 species that are currently known which use venoms. And I'm particularly interested in arthropods, so the creepy crawlies, which are like spiders and scorpions, but also centipedes or many insects actually have venoms like ants, wasps, and bees, or assassin bugs and robber flies, for example. So why am I particularly interested in the arthropods? Because as you can see here, the arthropods, they're actually the first land-dwelling organisms to acquire venom usage. So like scorpions and centipedes, they have evolved venom usage more than 400 million years ago. So these venoms are quite old and sophisticated. So they had a lot of time to fine-tune their venomous arsenals. And the arthropods, they're also incredibly diverse. Also, like of all the venomous representatives, more than half of them are actually arthropods, and they're understudied as well, because most of the research in regards to venom has been focused on, for example, snake venoms, because it's important for medical reasons. But I think arthropods are even cooler than snakes. And on the right side, I put a list what I have worked on so far based on my publications. So you can see in red are all the arthropod venoms, and I did one paper each on a cone snail and snake toxins, but it was sort of out of the order. So normally I'm focused on the arthropod venoms. And in general, what is a venom? So venom is an incredibly complex cocktail of lots of different organic and inorganic compounds. So I put the list here on the left. So most importantly, it's actually peptides and proteins and enzymes that are responsible for most of the activities caused by the venoms, but it can also be other components that cause direct activities or that help and synergize with the activity of the main compounds. And just to get a terminology right, so a venom, so we actually use the term venom if this cocktail is actively delivered, like through fangs or harpoons and stingers, like we have in spiders and snakes, cone snails, scorpions. But if a similar cocktail would be delivered passively, for example, like in frogs and toads when they're swallowed by their predators, then we talk about poison. So just to get the two terms right. And the term toxin means one individual component. So it can be one peptide, one polyamine, or one protein from a venom. And that component has to cause an adverse effect in the victim. So I put the proper definition below. Okay, so and how do we tackle this incredible diversity of venoms? So we use pretty sophisticated tools, which is like a high pressure liquid chromatography coupled to mass spectrometry. And here you can see this is a typical trace, HPLC trace of a tarantula venom. So we usually get about 50 to 100 peaks in the first step when we use a reverse phase column. And then when we further run each of the individual peaks through another step, in this case an ion exchange column, we might get another 10 to 15 peaks. And so this is from a paper which we published this year. So this is a venom from an Australian funnel web spider, and we analyzed it using 2 different mass spectrometry techniques. And overall, we found more than 3,000 peptides in a single spider venom. So this is incredibly diverse. And that makes it also very challenging to isolate individual components from this very complex mixture. And it also means that each peptide is usually present at rather small amounts in a venom. And so in order to do our research, I have actually started a venom collection. So I started in 2008. So this is usually going to Europe and collecting some of the arachnids that are kept as pets. And I've done 8 visits to Europe since then. And it was actually due this year, but then I had to postpone it due to COVID. So for the next visit, so hopefully I can do that next year to collect even more. So at the moment we are at about 550 different spider venoms and 150 different scorpion venoms in my collection. And this all came with a price because so this is actually my arm after collecting venom from some South American tarantulas. So they have a mean defense strategy, which is called urticating hairs. So they brush off some hairs from their abdomen, and they cause this urticating rash that lasts for a couple of days. But as the saying goes, no pain, no gain. And so this is sort of my publication output, the yearly publication output over my career. And you can see over the last 6 years, so this has increased significantly. And the red papers, they all come from, or they all had involvement of my venom collection. So you see there were some really nice papers coming out of that. So it was really worth going through that pain of collecting the venoms. And so what do we do in my lab in general with my collaborators on these venoms? So this is a schematic that I've taken from one of our recent publications. So this is general applications of venoms, and I'll show you what I have worked on so far. So a lot of the work that I'll do, especially with collaborators, is we are using specific toxins as molecular tools to study ion channels, especially sodium and calcium channels. We also look for interesting toxin components to target, for example, parasites. So my current ARC project is targeted at parasites of honeybees. We also looked into using toxins to treat certain medical conditions like pain, epilepsy, and irritable bowel syndrome. And one of the toxins was particularly useful to dissect the role of NEF1.1, a specific voltage-gated sodium channel subtype for mechanical pain sensation. I haven't really worked on the molecular probes yet, but a big focus in my group is actually on the bioinsecticides. So I actually worked with a lot of different insects and testing my toxins on them. And if you want to look, if you want to have a closer look at all my work, so all the papers are up on Google Scholar, so you can browse that website. So in terms of the labs that I'm planning to establish, so my LCMS system has been recently installed a few weeks ago, and this is really important to fractionate the crude venoms and also to purify recombinant toxins, because as I mentioned before, we have to make some of the toxins recombinantly, as we won't get enough to fully characterize them just by isolating them from the venom. And there'll be also my insectary, which is currently still in the planning stage. So this will be sort of one corner of the animal ecology workshop that I divide off. And so this is, yeah, just some images from the plants. And this is where all the arthropods will be housed and milked. I will set up some ex vivo assays, like some insect tissues like heart, muscle, and midgut to test the toxins. And I'll also have a range of in vivo assays testing on various insects that we use as targets, or like honeybees as off targets, to see that our toxins are safe for honeybees. And I'll also, so this is a new area of research that I want to dive into future. So this is looking into learning and memory in arthropods, and also how the toxins affect the learning and memory process. And yeah, so this is sort of my research in a nutshell. So I have basically, my lab has like three main pillars. So one is like the main research on the bioinsecticides and the antiparasitics, which is funded by the ARC at the moment. And I'll also do sort of testing for other people that have discovered interesting toxins, but very often they don't know what these toxins do. And I'll be offering them, you know, testing against insect targets, because very often a lot of toxins are actually targeted against insects. And the last pillar is sort of my arthropod venom collection, which I provide to various collaborators around the world who look into their specific areas of research. So thanks to my sponsors, especially to Roland, and making it possible that I can move to USC and giving me a good startup package, and to everyone from GeneCology for organizing this event today. And now I'm open for questions.
Thank you very much, Volker. That's great. It's fascinating work. I was just wondering, you say you go to Europe to collect spiders, yet Australia is so well known for its poisonous spiders. Do you also work on Australian spiders, or there's a reason why you prefer European ones?
It's actually probably the laziness, I would say. So Australia is also incredibly large country, and sometimes it's not easy, you know, to find the right spiders. You know, you might be 50 meters away from the collection site and you'll never see them. So you actually need to know where to look. And for my collection, I can only, or I mainly need larger individuals. So I can't really milk tiny spiders because they're just too small. And so the reason why I go to Europe is actually a lot of people keep, for example, tarantulas as pets. And there's many hundred species, and tarantulas are usually big, so they're easy to milk. And when they're kept as pets, they're also easy to source, because I just go to the home of that person who keeps them as pets. And in his basement, you know, he might have 100 different tarantulas. I just go to the tank, take one out, milk it, put it back, and then maybe 2 years later, I can come back again and milk the same spider, because they're quite long-lived. So they live up to 20 or 25 years. So that actually makes it a lot easier for me to get a really diverse collection, rather than going to all the different countries where they come from. But I also try to collect some of the animals in Australia.
Fair enough. I think that's great, and it's nice. Are you going to do some of your recombinant work here at USC?
Yes, that was the plan.
Good. That's great, because we're doing quite a lot of recombinant proteins, so it would be nice. Are there any questions for Volker? By anyone? I think I'm really delighted, Volker, that you have joined GeneCology. I think you will find collaborations growing as the time goes by. There's a lot of work, as you know, on peptides. I know Scott was probably the person who, through conversations, brought you over, and, you know, work on the peptides and work on the recombinant proteins, and certainly agree that not as cuddly as koalas, although I know that some people really love their tarantulas. So
yeah, I actually think they're cute, but
yes,
not everyone shares that opinion.
I have a PhD student who also thinks that. But, yeah.
Okay, I just had one question.
Yes.
I was just wondering, when you doing the recombinant proteins with the venoms, do you have a lot of trouble with toxicity to the bacteria and have to separate your proteins out to make them inactive while you're producing them, or?
Not so much toxicity to the bacteria, but what we have problems with is actually to get them properly folded, because many of these toxins are disulfide rich and they form disulfide bridges. And sometimes the system, like the enzymes in the bacteria that are responsible for folding peptides, they're not identical with the enzymes that are present in the spiders or scorpions. So they actually, we get a wrongly folded toxin, which then is usually inactive. So usually only one, like the right fold is the active one, and the wrong folds are inactive. But yeah, not so much toxicity to the cells.
Sure. Thank you.
Any other questions for Volker? If not, thank you very, very much for your great presentation.